Welcome everybody to Wednesday Night at the Lab. I'm Tom Zinn and I work here at the UW-Madison Biotechnology Center. I also work for the Division of Extension, Wisconsin 4-H, and on behalf of those folks and our other co-organizers, PBS Wisconsin, the Wisconsin Alumni Association, and the UW-Madison Science Alliance, thanks again for coming to Wednesday Night at the Lab. We do this every Wednesday night, 50 times a year. Tonight it's my pleasure to introduce to you Francisco Pellegri. He's the professor of genetics and the chairman of the Laboratory of Genetics, which is actually two departments, the Department of Medical Genetics in the School of Medicine and Public Health, and the Department of Genetics in the College of Agricultural and Life Sciences. This is my chance to say this building has three co-owners: the Office of Vice Chancellor for Research and Graduate Education, the College of Agricultural and Life Sciences, and the School of Medicine and Public Health. And that's rare that there'd be a collaboration between ag schools and medical schools on this scale. It's not too rare on this campus, though. There's two other buildings just like that. Francisco's going to talk with us about preserving biodiversity for the future. And Francisco, if you could come over here, I'm going to ask you the 5 questions.
Sure.
The standard rules.
Can you hear me in the back? Is it okay?
Is this audio okay?
The audio okay? No.
Good.
No, it doesn't.
So the rules of the...
How do you get them?
What's that? No, it's on. It's fine.
It's fine?
Yeah.
Okay. Go ahead.
I get to ask you these 5 questions. You get to answer them in any way that you would like. Francisco, where were you born?
I was born in Alicante, Spain, but I moved to Caracas, Venezuela when I was 2. So
And is that where you went to high school?
I did, yeah, Caracas, Venezuela.
And then where'd you go for your undergraduate degree and what did you study?
So when I was 17, I went to UC Berkeley for my undergrad. I studied genetics.
Genetics. And then where'd you go for your advanced degrees and what did you study?
I went to MIT for a for a PhD in biology. And then I did a postdoc in Germany at the Max Planck Institute for Developmental Biology.
And which city is that in?
That's in Tübingen, Germany, just south of Stuttgart.
Oh my goodness, the land of Immanuel Kant. Or is it Kant?
I can't hear it.
Oh, we're going to have to do, yep. Okay, so let's do a sound check here.
Can you hear me now? Okay, yeah, my voice doesn't carry very much, sorry. Plus I've been teaching all day. So I was born in Spain, south of Barcelona, Alicante, and when I was 2, I went to, my parents lived in Venezuela for 25 years, in Caracas, Venezuela, my parents, with my family, so I was there between age 2 until 17, so I essentially grew up there. That's where I went to high school. I was the last of my brothers. My parents wanted to go back to Spain, so I ended up joining my brother at UC Berkeley. He had a fellowship then at the time. That's why I went to Berkeley. I studied genetics there. And then afterwards, I went to MIT for my PhD. And then afterwards, I did a postdoc in the Max Planck Institute in Germany, in Tübingen, which is south of Stuttgart. And all of it was on developmental biology, essentially, developmental genetics, which is my expertise. And more recently, I've applied that expertise to trying to understand, trying to see if we could have some better tools towards, or join the fight for conservation biology.
And then when did you come to UW-Madison?
I came in 1999.
Great.
Mm-hmm.
Really appreciate it. I'm looking forward to your talk on preserving biodiversity for the future. Would you please join me in welcoming Francisco Pellegri to Wednesday Night at the Lab?
Thank you. Thanks, yeah. Thanks a lot for being here, for your interest. It's a pleasure to be here. Feel free to interrupt me at any time. I'm happy to just answer questions on the spot. In fact, that's a lot more fun. Does anybody know what this is? That's right. That's the seed vault in Svalbard, and that's has the so-called doomsday vault. But there's about a dozen or many seed banks of plants around the world, including some in Trinidad and so on. And of course, we all appreciate the importance of those vaults, of those banks, because they have seeds, and those seeds can help replenish biodiversity. And what I'm going to tell you about is more towards trying to use the same principle towards animals, which of course for animals we don't have seeds, but now we have different kinds of technologies. And even organizations like the San Diego Zoo or the London conglomerate 50 years ago understood the importance of this and already have been keeping banks of tissues for the preservation of biodiversity. So that's what I'm going to tell you about with a current outlook on it. And also with a pretty strong sense of developmental biology inserted into it as a way to get a tool to use those tissues and recreate animals, to recreate biodiversity. All right, so then now, as I said, I studied basic fundamental biology in early development using the fish as a model system. You may have heard the zebrafish is a very popular model system. Lots of labs study that. And I studied very early processes right after fertilization. They depend on so a lot of processes that depend on how the egg is made and how the embryo gets started. And I also teach. And in 2006, we have a capstone class that involves society, genetics and society. And a student, Amanda Gasper, who's now in Milwaukee, working for an organization that works with patients, she brought up this article. This article was it was in the Telegraph, and it was an attempt to resurrect the last member of a species that had become extinct. This was the Pyrenean ibex, and it made the news because it was the first of its kind. And it was also very poignantly sad because the one baby survived for 7 minutes, then it died. And it was also very poignantly sad because we really were not we were so completely unprepared for to use this kind of approach. So that's what got me thinking about the process. Just because I also worked on really early processes in development, so it overlapped my own interests in biology and my own interest in conservation, which I've always had in my life. So here's a little bit more history in the Pyrenean ibex as an example of what I'm going to tell you about is, you may have heard it, maybe not, but it's called the Extinction Vortex, which is where, unfortunately, the Pyrenean ibex found itself. In the early 1900s, it was recognized that it had been overhunted and that there were too few left, and that it was thought to be extinct, but a small subpopulation was found, so it was protected. A small population was found in the central Pyrenees, so this is between Spain and France. And but it was a small population, and it never recovered. So it was in 1989 there were only 6 to 14 left. In 1996 there was the last. There were 3, and 2 of them died, and there was one that had died. That one was still alive. Yeah, thanks. And so at that time in 1996, that was also the first time that there was the cloning of a mammal, and so that was Dolly the sheep, right? And so this was Dolly the sheep, and Dolly was born at first, and there were great hopes, and cloning is still great hopes. There was a period where people thought that Dolly was sick, she had aged too fast, but turned out that those issues have been solved, and that cloning is actually quite safe, at least after the first third of the gestation. And cloned animals are as healthy as non-cloned animals, at least those that make it at the beginning. And the beginning is more difficult because it involves reprogramming, and that always has inefficiency. But so Dolly was cloned, and so that actually gave the idea to the scientists and conservationists there in the Pyrenees to try to see if they could get a sample from this last ibex and get an ibex back. And that was what that attempt was. But they only had one individual, and so that's the individual. Even when there were a dozen or several dozens, the population was already what would be called the Extinction Vortex. What that is basically that when you have stressors and the population becomes very small, there's going to be compounding effects where you can have fragmentation of the population, and that may involve more inbreeding, And inbreeding will cause individuals to be less fit, so they'll have less progeny. That's going to reduce the numbers. There's going to be a loss of genetic diversity, and all of this is going to add up so that essentially it's all a downhill spiral from there. So that's what's called the extinction vortex, and probably and so that leads to extinction, and probably the Pyrenean ibex. The reason why it never recovered is because it was in that extinction vortex with that loss of genetic diversity. There's another example, for example, the Florida panther, which was essentially in the same path, and until there were several dozen less. But then they brought a panther from Texas, which was related, at least historically had been related, and that was able to rescue the extinction vortex. And so now the Florida panther is actually recovering, right? But in this case, they didn't do that. And so, of course, we know that the rate of extinction, this is just a couple of slides on just to show the rate of extinction over time is thought to be increasing. So this is from if you use fossils to track the rate of extinction, there's a baseline, there's always been extinction. But in the last 1,000 years, there's been this increase in extinction rates. And when you think about the absolute numbers, right now they're not that bad. They're around 1% or so in some species. I mean, in some different types of animals. But if you start thinking that critically endangered or vulnerable species, so the threatened species, if they also become extinct, that actually starts to look like a mass extinction, which would be essentially the mass extinction would be 75% of species that become lost in a relatively short period of time, which is a couple of million years, and we're really only talking about a couple thousand years in our case. This is the last slide for this is where if you start tracking down for the different animals, and these are the 5 different, the colors here represent the rates of extinction of in the five last mass extinctions. So mass extinction, again, 75% of species become lost. And so this one, for example, this Devonian, is when a lot of plants diversified, and in a kind of a relatively long period of time, they essentially overpowered the planet and changed the planet, so there was a mass extinction. And this Cretaceous is this brown is where the bolide fell on Yucatan and caused very fast the loss of a lot of animals, including dinosaurs. And if you standardize all of them to the same amount of time, so you just calculate it, so let's assume that they all happen at the same time, this will be about the rate of mass extinction. And again, if you use if you start thinking that the threatened animals, with good reason, the threatened species right now are going to become lost, really the rates that we're talking about in current time are very close to a mass extinction, if not there, if not faster. So if all critically endangered species become extinct in 100 years, it's been calculated the time to mass extinction will be between 1,000 and 2,000 years. And if you even include the ones that are now threatened, which is the lowest level of threat, that would actually be between 250 and 500 years. And we know that there's kind of a perfect storm of situations with planet having reached a certain mass in the planet and the climate change this could be a perfect storm to create conditions with a lot of stressors. So my job as a developmental biologist has been to try to think of ways in which the cloning of that Pyrenean ibex could have been better, could go forward, and techniques that could allow samples to use methods that could recreate an animal from a sample, just like you could make a plant from a seed. And there's 3 basic methods I'm going to tell you about. The first one is cloning, which is what they were trying to do with Pyrenean ibex. The other one is germ cell transplant, which is basically you make a germ cell, you use a host to harbor a germ cell. And the last one would be a whole animal chimera, which is basically you make a chimera of an animal, let's say a cheetah and a cat, and you could get gametes of a cheetah, for example. And so I'm going to tell you a little bit about that. So what about cloning first? This is an example of the Pyrenean ibex. This is cloning. Cloning is this basically, and this would be for Dolly, right? So it would be for Dolly, you'd have a sheep, and you'd clone a sheep using a sheep egg. And so the basis of cloning is that you take a nucleus from a somatic cell, it could be a skin cell, for example, or a blood cell, and you put it into an oocyte where the nucleus of the oocyte has been removed. Then you have a reconstituted zygote right there, and that can grow into an embryo that can grow into an adult. That's cloning. What they tried to do with the Pyrenean ibex it's the same, except it's called it's interspecies. That's because they had no eggs for the Pyrenean ibex, right? So they had to use an egg from something else. And so here the egg came from a related species, which is a goat that they used. And they used the goat because it was easier to get, right? Of course, that's simple. That made it simple. And the nucleus was from tissue that had been frozen from a biopsy from the last Pyrenean ibex. And so here, what you see is that the green nucleus has to be able to work with the blue egg, essentially, right, for that to work. And that may not be the case because these are different species, right? So what we'd like to understand is, does this work? Would it work? When would it work? And if it doesn't work, could we make it better? Turns out there's some examples where it has worked already. So here's an example. This is a mouflon. This is a wild goat from Persia that is endangered. And this study was done. This was in 2002. It was the first example. And they used the egg of a sheep, an enucleated egg of a sheep, and the nucleus of goats, of the mouflons, and the mouflons were dead. They were dead in pasture. The cells were dead. The nuclei still were okay. Even though the cells were dead, the nuclei were still okay to clone and get a mouflon using the egg of a sheep. It turns out you can clone an animal even with a nucleus that has been completely freeze-dried. So that might be a nice way to store them, perhaps. Here's another example. This is actually, if you look at the felines, and it's a lot of fun, there's lots and lots of species, lots of cute patterns. This is an example of a wild African cat, and it looks just like a regular cat, but it's a different species. And it also has been cloned using a domestic cat egg, which is a different species. So another example, if there's species clone. You can clone a wolf from a dog with a dog egg. That's not super surprising. They're actually the same species, except one is derived from the other. You can also clone a coyote using a dog egg because they're very related. Here's another example. This was done by recently. This is in 2020, using a sample of a Przewalski horse, right? So this endangered wild horse, which was fortunately rescued through captive breeding and survived, and now it's running loose in Mongolia. But the genetic diversity of the Przewalskis is not very high because they came from a really strong bottleneck, and so they had lost their genetic diversity. And genetic diversity is really the health of a population, right? If we think about COVID, for example, the reason that, as a population, we're okay with a COVID is because there's so much genetic diversity that some people are not responsive to COVID at all. And in fact, there's a recent study in NPR, that recent study that was highlighted in NPR, that they looked at that it looks like people who survived the bubonic plague actually carried the better genes that would allow us to survive. So that genetic diversity is actually what allows us to survive a population. It's the same for any population of animals. So genetic diversity is essentially the health of a species. And so now this curd, this is from a nucleus that was frozen for 40 years at the San Diego Zoo. And got a baby, and the egg was from a quarter horse. And so that was an example of interspecies, but they were actually subspecies of a successful one. Here's one that was even more recent. This is Elizabeth Ann. You may have seen this in the news. It was a big deal. So the black-footed ferret is an endangered species from the United States that also suffered a very strong bottleneck in the 1970s. And they tried to start a little group that they could breed. And so those were bred and eventually released back into the wild, but they are really lacking on genetic diversity. And they had 2 samples that were frozen again for 40 years in the San Diego Zoo. And they were able to get, one of them actually has canine distemper, so they cannot use that one. But the other one, they were able to get Elizabeth Ann to survive. And she has 3 times more genetic diversity than the entire population that is existing. So she really could reinvigorate the population with genetic diversity. Okay, No, so now this kind of I forgot to say. Elizabeth Ann was actually cloned using a nucleus from a black-footed ferret, which is the American species, and the egg was from a European ferret, which is the domestic ferret. And those are separated by 1 million years apart. Those have already been separated 1 million years, but they are two different species, closely related but not the same. Again, it's an interspecies cloning method. All right, so then it's clear that if you're close enough, you could maybe get this to work, right? Because from a developmental perspective, the developmental pathways are compatible. But as you start going further and further apart, and there's literally examples in which people put a nucleus of a whale into a pig or a panda into a rabbit, that's not going to work, because that's too far apart. And so the question is, how do we know how far we can go, right? And and how can we capitalize on that? We really don't have time with just the 66,000 species alone to figure out everyone. We can actually make, from a cell, we can make gametes for a mouse in vitro. No animal at all. We can do it all in vitro. But we don't have time to figure that out for all 66,000 species. But if we could use one species helping the other, using conservation pathways, that may help make it happen. So how do you draw the line? So I mentioned that I work on fish. And so what we did is there's several thousand labs studying zebrafish, and there's about 2 or 3 labs in the world that have more than just zebrafish. So what we decided is to use, to look at this tree of the related species in the fish and take advantage of everything we knew in the zebrafish to try to understand how far we could go using this kind of approaches. And so that's, right now we have we have at least we have 8 or 9 species in the lab, which are very fun to have also. Right. And then the other thing about it is that this also was something that was part of my research. I don't have time to explain it, but there's these methods that were developed in the fish. They were originally developed in frogs, but they have been applied in the fish. They're very helpful for genetics study, but they're methods they call Ploy Manipulation. But they're basically methods where you can just generate an offspring with either the maternal nucleus or the paternal nucleus alone. Okay. And I'll give you the example where it's the paternal nucleus alone. Okay. And the way to do that is you take an oocyte. So you're familiar with in vitro fertilization, right? You get an egg, you take a sperm, you fertilize it in a petri plate. So this is the same thing. It's not cloning. Cloning is really hard. You need a needle, like micro manipulation, But this is very, very simple. What you do here is you use irradiation, and we go to medical physics for that, but there's easier ways to do that. And you essentially damage the DNA from one of the gametes, in this case, the oocyte. And then you use the sperm, and then you introduce the nucleus with the sperm. So in this case, the sperm is only paternal. Does that make sense? Okay, so that embryo is only paternal. The only problem with that embryo is it's a haploid. It only has half of the chromosomes. Okay, but we also have ways to actually duplicate that. I'm not going to get into that, but we have ways to duplicate that. The way to do that is simply to inhibit one cell cycle so that the DNA replicates and the cell doesn't divide, and it just changes from half to a full amount. And they actually survive to adulthood. So we can very easily, or at least routinely, not easily, but routinely we can get fish that only have maternal DNA without a father, or fish that only have paternal DNA without a mother. And those are methods we've been using for a while. So we thought, okay, let's use the same method, now trying to mimic interspecies cloning. Right? So instead of doing the hard work, which would be cloning, we'll just try this blue and green, do they work question, using this very simple method. Okay? And so we basically can take a different species, introduce the nucleus of a different species into the egg of a host species and ask, does that work? Does that make sense? Yeah.
Is that genetically identical to the father then?
Yeah, right. So
why is that not same as a clone?
Oh, I see.
Genetically.
Yeah. Well, there's two things about that. The first one is not a clone because it's not a cloning method, and a cloning method means you literally move it and transplant it. So that's very difficult. But it's actually not a clone because from a different perspective, from a genetic perspective, it's not a clone because that actually has gone through meiosis and it has scrambled its genome with recombination. So, but that doesn't matter too much because what really we're testing is even if the genome is scrambled and one sperm is not the same as the next and so on, what we're really testing is does the green work with blue here? Does that make sense? Yeah. Okay, great. Good. Nice question. Thank you. So then, now what happens in evolution is that we may have an ancestral species, say the ancestor of the ibex and the goat, and then at some point they diverged, right? So as they did that, they started evolving separate from each other. Now it turns out that there's components in the egg and in the nucleus that need to work with each other. And so what happens if you now try to do this later, those components may not work. And what we're trying to do is what are those, we know about some of those components, we're trying to test some of the main components. And so the first one is that you may have heard of the mitochondrial Eve, right? I mean the ancestral Eve, right, in the news. Well, that's because we can track mitochondria, and mitochondria are only inherited through the eggs. That's how we can actually track the ancestral Eve, because the mitochondria only come from the egg. So here, if you use an egg from the blue strain, then that's going to have mitochondria. And the mitochondria has DNA, so it has some genes, and that DNA needs to work with genes that are now in the nucleus, and that's essential for energy production. Okay. So normally, when this splits, the mitochondria may change. The actual mitochondria change kind of fast because they're originally from bacteria, and their DNA replication machinery is kind of inaccurate. So the mitochondria may be changing, but the nucleus is catching up, and they're always correcting each other. Right? So they co-evolve separately, but then later when you mix them, they may not match anymore. That's what's called an incompatibility. Does that make sense? Yeah. Okay. Great. The other thing that is really important that is really important to think about is that the green nucleus is going to have genes, and the egg, one of the functions of the egg is to start the genes in the nucleus to get going. That's called the activation of the genome of the embryo. And that depends on transcription factors in the egg, and they have to recognize the sequence to activate the genes. And that also has to match, and that also may have changed. So anyway, right now we're looking at using the zebrafish to try to there's other things. For example, transposable elements have to be kept in check. And if you don't, they may start jumping around and affecting the embryo and so on. So there's other things. But those are two main things. And so we have students looking at those parameters. But I'll tell you a little bit about just the outcome. So remember that I told you we could get an embryo that was a haploid, but it's looking pretty good. Here's a zebrafish embryo haploid that is essentially the nucleus of a zebrafish from the sperm into the egg. So this is our control. This is what they should look like if everything's okay. All right, so now we're testing the next. Now we're testing a nucleus from this related species into the egg of a zebrafish. It's still okay. So this is kind of like the black-footed ferret in the domestic ferret egg, right? It's still okay, it still works. So those are compatible. And then the next species down that we've tried is Margaritatus. Right here, that's not okay, it's not growing so well. And so then now we have a way, first of all, we know that within a certain parameter, it works. And then we know that there's other parameters that are too far apart, as we expected. At some point, it's not going to work. But we can also ask, why doesn't it work? And can we make it work, for example? For example, maybe the mitochondria has changed. Maybe we could gene edit the mitochondria to make it work, and then it would work. So that's the gist of what we're trying to do. Now, this is, so we have the compatible, the incompatible group. from a scientific perspective, most of our science is actually there. Now, the long-term goal is what I can tell you a little bit more about. But please feel free to interrupt me. The other thing we realize is that there's lots and lots of tree branches in the tree of life, and this is just one of them. And evolution is just kind of random. There may be some kind of order in the way that the clock ticks, in a way the clock of changes, but we could really be fooled by just looking at one lineage. So we had to look at various lineages, and so we also have some frogs that are from Madagascar. And the important thing is they're all very highly related, and we're hoping we can do something similar. Right now, we're just trying to breed them. Some of them are endangered, but they breed okay in the wild. So those are poison frogs from Madagascar. They're nice to have, and so we'd like to do the same. We also work with students in a class. I actually teach a class year-round with students, and it's almost like the Innocence Project, where we're always trying to work on a project. And I know we don't get as close as actual cases as in the Innocence Project, but they're always thinking about this. And in one of their projects, they identified dolphins. And look at the dolphins. There's about 6 of them that are less than a million years apart, species of dolphins. And that's really close, right? So we probably wouldn't work with dolphins, but we could work with cell lines from cetaceans and kind of ask the same kind of questions, and potentially the same principles could apply to that lineage and other lineages. All right, so then now I told you a little bit about cloning. Cloning is rough on embryos, because a clone comes from a differentiated cell, and the differentiated cell has, when an embryonic cell starts, it has the information, the DNA. It's like a library with all the books are there. The information of the entire library is available to you. As the cells start to what's called differentiate, they will choose paths. For example, some will be heart cells or muscle cells. And within the blood cells, some of them are going to be white cells, and some of them will be red cells, right? As they do that, essentially the genome or the cell is essentially taking books away and cordoning them off. The ones that are not needed are taken away. The way it's done is by actually compacting them. And that's what's called chromatin organization. So they regulate at the level of compaction. And that's done by modification of the DNA and the proteins that bind the DNA. So a differentiated cell has lots and lots of those changes in it. And then when we do cloning, we're asking that cell to go back to the nucleus and suddenly open up, or take away all those cordons and open up every single book in the library. And that's really hard to do because the embryo is not set up like that. There has been no selection in evolution to allow an animal to be cloned, right? So that's something that we're doing. So that's why there's quite a bit of attrition for embryos during the first trimester of gestation, or the first third of gestation. Once they pass that, they've actually made it, and they seem to be pretty healthy. But there's more subtle ways to do what we're thinking about doing. For example, this is my favorite. This is, I think, in the future, maybe this is what is going to be the best way to do it. This and the next one, actually. This is called germ cell transplant, which basically you could take germ cells. Now, the germ cells are the precursors of the gametes. So the first decision in an embryo is whether a cell is going to be somatic, and that's just going to be the organism, or the germ cells, and that's going to make the gametes, either the sperm or the egg. And the germ cell is basically what's left for the generation, for the next generation. Right? So, and we know we know how to identify germ cells. We start to understand how to make germ cells, and we can isolate germ cells. Even in birds, it's actually pretty easy because they're pretty big, so you can actually make bats of germ cells isolated. And you can take germ cells from one animal or even from a petri plate and put them into another. Here's an example where they have put cells from a black chick line into a yellow chick line, and you see those little black patches. Those are cells from a black chick line. But if you looked at the germ cells, that chick may also have some germ cells from the black chick. And so the idea is that we might be able to get a somatic cell, and we're starting to understand how to make germ cells, and we could put it into an embryo, and we could now have another embryo carry those germ cells. And this embryo could be from one species and the germ cell from the endangered species. So using a non-endangered host to carry the germ cells of an endangered species. Does that make sense? We already know, for example, that a chicken could carry quail germ cells. So we know that a chicken could grow up and actually make gametes that would be from a quail. And so that might be an easy, and that chicken would be really unaffected. Now this is actually the same thing, except that rather than manipulating the germ cells, we're manipulating the entire animal. And this is pretty applicable in mammals, because mammals have this early development that is very plastic, so essentially they can absorb cells into the organism, and it doesn't affect them very much, because mammals have a very plastic early development. And it's called whole animal chimeras. And so here's a graft, right? And so we're all familiar with the graft in a plant, right? And if we have a graft in a different plant, but you get gametes, you get pollen, right, or seeds from the part that has been grafted, the seeds are going to be from the grafted plant, right? It's going to be from the sector it's going to be the sector that gives you the gametes, even though it's in a different plant. So it's kind of the same thing, where you could generate a chimera, where you take cells and you put them into an embryo. And again, mammalian embryos are very plastic, so they absorb cells very easily without affecting their development. And you can now make whole animal chimeras. These are also used for biomedical research. For example, a lot of people are thinking about making hearts, for example, in a pig. But this is a mouse, and this is a rat. Okay? And this is a rat carrying some mouse cells. So she's a chimera. And this is a mouse carrying some rat cells. Can't carry too many, because otherwise it would reject it. But it can do it, and they're healthy. And just as you see the outside, the germline might be the same, and so it could also carry gametes from each other. Yeah. And so here's an example of an article that from a different review. In this case, they have an endangered rat from Japan, and the idea is that you could make these chimeras from a mouse, using a mouse because it's related, and then you could get a chimeric between the two species, and then those chimeras, if it's a female, it could get eggs from the endangered species. If it's a male, you can get sperm from the endangered species, and if you can isolate the gametes, you can literally recreate the endangered species. Or, for example, a cheetah and a cat, something that you could potentially get a chimera. And of course, nobody wants to make a chimera necessarily, but if it allows you to recreate the species, or it allows you to keep that species healthy, it starts to become valuable and worthwhile. All right, so then I told you about these three different methods. They all involve interspecies approaches. And they're all a little bit strange, right? It's an unusual step. But in all cases, the beauty about it is that in all cases, you really can get to a regenerated individual that has no changes at all from the original species. And that's what we want to do, is be able to recreate individuals without changing them at all. Yeah. Does that make sense? Any questions? Yeah.
I guess I have a question about
Sure.
the whole philosophy of this project, because I understand that species are going to be extinct, and that can be the result of disease or climate change.
Mm-hmm.
And so I'm trying to understand your vision in terms of having a biobank of just different tissues,
Right.
in terms of biodiversity,
Sure.
of what it looked like 100 years ago, for example.
Mm-hmm.
How could this be used in the future?
Right. Yeah. So the idea is that, so the idea is that you would have these banks of cells as a backup in case you need it, and just like the future is is that very likely, in many cases, there's going to be stressors in almost all ecosystems, and that as soon as you affect one species, you might affect more, right? Because they're all interrelated. So the future is you could actually monitor biodiversity, and if you have the samples, you can actually reinvigorate the diversity. But if you don't have the samples, you could never do that, and you lost the opportunity. And now is the opportunity. And if we don't need the samples, great, right? Obviously, obviously making ecosystems healthy now is what we need to do. But if we have, this is not necessarily detracting. It's a backup mechanism that could really help keep species under stress, which is what has been predicted for the next couple hundred years at least. Yeah, I'll get into that a little bit more. Yeah. So let me get into that. Great question. I'm completely with you there. But this is how we can contribute, right? And so we can talk more about that. I'll try to wrap up quickly so we can have more discussion. But anyway, so the idea is that, and this slide is from the San Diego Zoo, is that you can have endangered species biobanks, and then the old biobanks was basically saving eggs and sperm or frozen embryos. And you can imagine that's highly invasive, right? And you have an endangered species, you're trying to get their eggs, that's not so great. But now we really have the ability to reprogram cells. And that's what the beauty is about, is that you could literally get hairs that have been pulled out in a hair trap, and that could have cells that you could actually, using molecular methods, turn into reprogrammed cells, grow them in vitro, and freeze them. And it's completely non-invasive, right? And so those are the kinds of methods that come in. And then if we also have the ability to turn those into gametes. And the important thing is to really save the information, because once the information is gone, it's just gone, right? So, yeah. So covering the tree of life. So the idea is that kind of trying to allude is that you might be able to have, and this is where the development of genetics and phylogeny comes in, that you might be able to preserve species in groups, in clusters, because they have conserved pathways that you can piggyback on each other. A simple example is that the northern rhino essentially became extinct, but they're thinking that they can get some back by using a southern rhino as a surrogate mom, because they have frozen embryos, but they don't have a mother, but they can use a different subspecies as a surrogate mom. So basically, these clusters of species might be able to help each other if needed. And then we are learning more and more about how to make changes. For example, well, the technology might improve, and we may be able to cover the tree of life in this way better. And this could be applied to endangered and non-endangered pairs of species. Or it could be applied to simply species where there's a captive population and an endangered population in the wild. So, for example, the axolotl, it's nearly extinct in the wild, but there's lots of axolotls all over the world as domestic species. Now, this could not survive in the wild because they've lost the ability to live in the wild, but the nuclei from this could be going to the eggs of these and actually recreate the population and make them and grow more axolotls that could live in the wild, because those are wild nuclei. Same for dromedaries. Dromedaries is another example where there's very few left that can survive in the wild. All right, so then the biopreservation, there's this, what would be the rules? First of all, there's this, so I mentioned that it was very sad about the Pyrenean ibex because it was already in the extinction vortex. So you want to make sure your number of samples is not in the extinction vortex. So there's this rule called 500-50-500, which basically means that for short-term survival, you need 50 individuals. For long-term, at least 500. So we want to biopreserve, we need to do it now and do at least 500, 1,000 samples of different individuals. This is the passenger pigeon. The passenger pigeon, there were millions in the sky before it became extinct, right? And after overhunting, and because they had a certain life cycle where they needed large numbers, they became extinct. So we really don't we really don't have a sense that we don't have to wait until any species could become extinct. And so, and also there's, this is the fires in Australia, where large amounts of animals became affected, and so we need to have the genetic diversity before those events so that we don't lose the genetic diversity. And so the idea is to biobank proactively rather than wait until a species is extinct, to just biobank them now while we have the genetic diversity. It would be the easiest to do, and we are able to do it. Non-invasive, again, really important that it doesn't affect the population that's existing, so we know now we can clone animals using hairs, you can clone animals with tiny amounts of blood. It can be coupled to field study. We can even get blood cells from potentially blood-sucking animals. Like this is a mosquito, and this student was at the zoo, and he was able to get rabbit, deer, fox, and bison in mosquito blood meal at the zoo. So basically, there's multiple. You can get cell samples from fecal samples in the wild, and then you could get biobank. There's various ways that we would be able to get non-invasive samples. And you also want it to be sustainable for the long term. So a lot of those samples were frozen for decades, right? The black-footed ferret, the Przalski, that was 40 years frozen. So you don't want to have all this like whole building of freezers, and then somebody decided this just costs too much money, we never use it, why, right? So the more effective you can do it, the better. And so we are also exploring that. And for example, here's an example of this, I don't know if you know, but this little animal is called a water bear, a tardigrade. He has survived all mass extinctions. It can survive in space. If you dehydrate it, it turns into this little ball. And so we're in my lab, and other labs are studying how that happens, and could you protect cells with the genes that are expressed in a water bear, to essentially make an animal cell kind of like a plant seed, so to protect it. All right, so I'm pretty much done. I just told you that we think that it would be important to do proactive preservation of biological samples now while we have the genetic diversity. That somatic cells make it much easier to gather samples non-invasively, and that reprogramming of those somatic cells can turn those cells into gametes so that we can actually make individuals to regenerate an individual to reintroduce genetic diversity, just like with the Florida panther, for example. And that works best if we understand the fundamentals of biology. So anyway, I think that overall, to get to your question, is that at some point we like to be at this point where we know we're going to have some reserves and some areas that are protected. And but even there, like for example, in Costa Rica, which is highly protected, right? But the cloud forest. The cloud forest forms because there's an interaction between wind from one side and wind from the other, and climate change might just obliterate that regardless of how much Costa Rica protects it, right? So there's a lot of stressors that are really kind of uncontrollable, and so but having this tool, even though of course ecosystem preservation is the most important thing we have to do. Right now, there's different kinds of funders can also provide support for a different kind of approach, which is a little bit more reactive, which is where you might foresee a decline in genetic diversity and then and then essentially change it so that you don't have that decline, and therefore preserve ecosystem health. And so that's it. Yeah, I'm happy to have questions. There's a lot of students that work in this project through the years, and they learn about this in class, and we're actually going to Costa Rica in 3 weeks. And we also work with the Nelson right now, and we've had support from campus on this, with people in bioengineering, with tardigrades and entomology, and so thank all those people. I'm happy to take questions. Yeah, thanks. I know this is a topic that has a lot of kind of discussion. Yeah.
When you are using a nucleus from another cell into a enucleated egg of a non-identical species cell.
Yeah.
Why not, if you can bring a frozen nucleus why not bring the frozen mitochondria as
Ah,
well?
yeah. No, that's a great point. Yeah, why not bring the frozen mitochondria? And that's the kind of thing that we'd like, that's the kind of thing that we're looking forward that we need to do. Exactly. That's right. So you, for example, Elizabeth Ann, right, the ferret, she is healthy, but she's not reproducing, right? So is she not reproducing because her mitochondria is actually from a European ferret? It actually would not be a problem if she was a male, because males don't have, well, in terms of at least passing along, because they don't pass the mitochondria. But anyway, but yeah, in general, we could, there's ways, there's also this mitochondrial replacement for humans, right? Where you can take another oocyte that has the mitochondria as the donor for human patients that have a mitochondrial disease. So you could do that, something like that, right? You could, to rescue it. Or you could, I mean, there's already people at Princeton University making synthetic mitochondria. And that's a much smaller structure, potentially can be done. You know, so that those are the kinds of things that even having species like the zebrafish, we can test how to do that. Exactly, but that's exactly the right thing to be thinking about, yeah. Now, if we have a biobank and we can just turn into germ cells without using a nucleus, without using cloning, we don't even have to think about that because the mitochondria are there already. But if we had, say, let's say a dried up nucleus, then we probably would need cloning. So for some methods we might need cloning, for some we might not, and so there's different challenges. Yeah. Thanks. Yeah.
We have a question on Zoom.
Oh, we're on Zoom. Oh, great.
Yes.
Okay.
Don't you think we have to consider reinvigorating endangered prey as well for each species?
Endangered prey?
Yeah.
Yeah. Well, I mean, we know that, you know, the ecosystems are a network, and everything has to be, you know, the network works with at all trophic levels, right? And so the way to keep the ecosystem healthy is to keep all the trophic levels, regardless of whether it's a prey or a predator. I mean, and so the idea is to try to keep all species with enough genetic diversity and healthy, and not discriminate whether one or the other. There's a really nice study on the sea otter in California, and how it was protected and it came back. And they show how much it helps. There's a lot of studies that show that whenever the animal that used to be in that ecosystem is reintroduced, it really helps the ecosystem get back to what it used to be, and that involves forager plants and prey and predators. So all those interactions are important. Yeah. So yeah, hopefully that addresses the question. Yeah, but it doesn't mean to discriminate prey or predator. Yeah.
So it's highly cool science. You didn't tell us about how you reprogram nuclei.
Sure. Yeah.
And then I have a follow-on.
How do you? Yeah. So the reprogramming of the nuclei is something that was started. So there's the Nobel Prize in 2002 was given to this work, and there's these factors called Yamanaka factors, and what they did is basically you can make stem cells, embryonic stem cells from an early embryo, and they found the top 40 most active genes. And that just by elimination, they found that with 4 of them, they could reprogram a cell. Those cells are called induced pluripotent cell, and that just changed the field. So they can take any cell and make with those 4 factors. And so, and those 4 factors you can introduce just with a protein. You don't have to introduce a gene, so you never have to make a transgene. The nice thing about it is that once they introduce, they actually activate its own genes, so you can turn you don't have to make a transgenic to turn on its own genes. So it's basically a transformation with these factors.
Yeah, I thought so. So then there's philosophical questions.
Yeah.
How do you decide which animals are going to be saved?
Exactly, right.
And if the environment has changed to cause the extinction, which is caused by humans,
right?
When you reintroduce the animal, what's the likelihood that they could actually adapt to the changed environment anyway? Is this not a self?
Right. So I mean, yeah. So that's a great question. You know, the question of prioritization is really key, right? And a lot of times right now we think of an animal as charismatic, right? And those are the ones we care about. So we were talking with Paul Robbins. He's a bioethicist. That's a question he asked me also at one point. As I say, I don't know. I mean, this is from a technical perspective, this is what we can do. And then I brought up something about, well, maybe it would be nice to test like a contained ecosystem like Madagascar, where all the animals are related, and you could actually prove that you can do it at that ecosystem level. And there is a land-based ethics, so you could say that you're prioritizing an entire ecosystem rather than as individual species, and that would be there's a precedent for that as an ethical principle. And so what we're trying to do right now is doing that in the Great Lakes area. And of course, that's where we are. There's other ecosystems, and ideally this can be done in other places, of course, at the ecosystem level. But that would be that's what we're trying to do. Yeah, and so how do you do that? Well, you might start with the upper trophic levels, so to the more, but then go to the bottom, and then when you get to bacteria, maybe you have to store it a different way, but you might be able to do it. And what we want to do is show that it can be done in a small place. We could start in the Catalina Islands or somewhere small, but show that it can be done and then just expand. Does that help? Thanks.
Question here in the back.
Yeah.
So I have a science storytelling question about, sometimes they refer to your topic as the sixth extinction. So how do you introduce this to the lay public of skeptics so that it doesn't come to that? It's difficult enough to talk about the scientific definition of global warming versus climate change. People don't understand the scientific definition. They think that climate change is better than global warming. And so with that issue, how do you talk about the storytelling of the sixth extinction?
Well, I mean, there's a great book about the sixth extinction, got that Pulitzer Prize, right? That's the book that I refer people to read. I think that it's a reality, right? And so whether I think as a whole society is becoming more accepting of the idea that climate is changing, and where we come from is the perspective that because of those changes, there's going to be a lot more stressors in the system. And those could just be like really random events like extreme heat that causes major fires, right? And so that's what's going to cause all these double bottleneck effects on animal populations. And so we come from the perspective that given all this and that we essentially, this is going to be a given, let's just biopreserve as much as we can because the technology is going to be there, but we're not going to have the tools. And the other thing that's kind of relevant, I think you have a follow-up relevant to this is even when, I mean, there are studies that show that even when you try to, let's just say geoengineering, right, where you try to stop the ozone, try to stop the heat with particles and so on, there are studies that show that even there you're going to have even larger changes on the population that may not necessarily help the extinction species because the changes are just too fast. So yeah, we come from the perspective of trying to maintain the genetic diversity and of course trying to work globally with everybody else to change climate change. Yeah, sure.
Well, a persuasive part of your talk was the acceleration of the extinction and showing over time and trying to make that graphic as persuasive as possible so people can understand that and
sure.
Clearly looking at it.
Yeah. Well, so that's work from somebody else. I actually never use it. But because it was a broader talk, I'm using it. But I think from our perspective, extinction is happening, and so maybe I have to emphasize that more for the public. And then, but from our perspective, what we're trying to focus is on all those innovative or new techniques to try to counteract rapid changes in the amount of diversity in a population. Of course, if we can make people more aware of changes, so that's one thing we do. I take students to Costa Rica, and I teach year-round, and this is an important talk, and we try to do as much as we can. Work with the Nelson Earth Day at the Nelson Institute. This year is going to have the person that they cloned, the black-footed ferret, as a speaker, and we're going to have a workshop to try to work out all this innovation applied to conservation genetics. So yeah. if you have ideas, I'd be happy to
There's
appreciate
another question on Zoom.
that. Yeah.
Have you tried ChatGPT to see how good it is to get answers regarding reinvigorating biodiversity?
What did you say? What thing? AI. AI. Well, I mean, there's a lot of modeling, right, going on, and I'm sure that involves AI in all places, starting from climate change predictions to biodiversity prediction. From our perspective, so I'm a developmental geneticist, and I'm sure that that's starting to get into, for example, the reprogramming world. But we really get into what I'm proposing is working with living species. What we're proposing, it's not just me. There's people at the San Diego Zoo has been doing this for 50 years, right? And London Zoo and others is really work with the living species and try to preserve biodiversity. But for example, there's I don't know if you've probably heard that there's this company Colossus, right, which is a splinter of these black-footed ferret people and the company. And Colossus is the one that's doing the mammoth, right? And the mammoth de-extinction, and that's a long time. That's not what I'm talking about. I'm talking about living species, but those are long lost species. The Tasmanian tiger, which is not that long. We actually have photographs of the Tasmanian tiger, right? Dodo bird, and the passenger pigeon. And they're trying to reconstruct an entire animal with very little sequence. So I told you about the 5,500 rule, right? So we want to have 1,000 genomes. So it's easy for us to do things. But how do you then make an animal and reintroduce diversity? We just have 1 or 2 partial sequences, right? And that's really a challenge. I would say almost impossible challenge to do. And there they're actually using AI, because what they're trying to do is compare. I was talking about this, how the DNA was packed and all that, and they're trying to understand really what are the sequences out of this 10 billion of DNA bases, which ones are the ones that are important to make a mammoth, right? And there they're using methods like that. Yeah. But what we're trying to do is much more humble. It's essentially the diversity is there. All we want to do is save it now so that people in the future, 50 years from now, who's going to have incredible techniques are going to be able to use it. Yeah.
Well, to follow up on that question is how much of your data is in the cloud, is digitized, because this Snapchat that they're talking about is looking at the myriad of what we have on the internet, and the amount of data that you collect is pretty insurmountable. And in fact, I think there's going to be a convergence of digital electronic data storage and DNA storage when you talked about that one type of animal that is so non-destructive.
Yeah.
That you can use that as a method of storing all of our data.
Yeah, so there's a lot of efforts to get DNA information from a lot of species. And I think, hundreds of years from now, maybe 1,000 years from now, I don't know when, we'll probably be able to make an animal with just that. But we're not there yet at all. So we really can't recreate an animal from scratch. We really need an egg, we need a nucleus, what's going with it. So in the timeline of this predicted extinction, information in a microchip is not going to be very helpful, I think, at all. It might be helpful to reintroduce diversity with CRISPR, for example, but that's not. So what we're talking about is banks of physical nuclei, and for that, yes, I think we should have as much open access as possible, and we're trying to work on that kind of network with other people who want to do the same. Yeah, yeah.
When you go to Costa Rica with your students, are you also doing genetic surveillance? Where you are monitoring within a population of species?
Mm-hmm. We're very early in the stages. All we do right now is just a fun exercise to try to catch mosquitoes. But eventually, yes, the idea is to do that surveillance. But right now, we're just trying to kind of get, also gain their trust, because these samples are theirs, and we don't even have to take them. All we want to do is make sure. Same for, we also have a collaboration in Nepal, which is the same thing, and the samples can stay with the local community, which is completely fine unless they want them to be in a centralized place. And so all we're trying to do is understand methods that we could catch samples. Now, we were there, and they were super excited about a little patch from a tapir, and they found the marks on the ground, And they're so excited and measuring it. And so these are animals that you don't even see, right? They don't even see, even though they live there. So how do you catch samples from them, right? So that's where the mosquito, but other people have used mosquitoes before for genetic analysis, even in villages in Africa. So that's where it came. And even the London Zoo has some mites as repositories of bee DNA and so on. So it's not our own idea, but it's a fun idea. So and next time we might, add fecal samples in Africa, and the idea, and hair in tigers in Nepal. So the idea is to just be non-invasive, but ultimately also coupled with field studies, because that's probably going to give us a lot more. But just tiny amounts are enough for us. Right now, what we're trying to do is use samples from the wolf hunt and try to set up a pipeline where. And I was just meeting with people at the Weizmann today, actually, because they have an IPS induced pluripotent cell. So what we'd like to do is take samples from wolves that have been hunted because they're there, and we can make cell lines from those. And then we can have 50 and then mix them, put them in a tiny little capillary that represents 50 individuals. Freeze it, and then see if we can bring those back, for example. So that would be kind of the principle. Yeah.
One last question.
Yeah.
I actually have a question regarding the Dutch Tourist Foundation program, where Dutch scientists brought back the original wild cow in Europe.
You mean the
and there's
tarpon
actually,
horse?
Sorry?
You mean the tarpon horse? Oh, the auroch?
Yeah, the Ark.
Yeah.
And it's been alive for the last 7 years. So I was wondering, so all the stuff they learned, could that be used for the ibex or the sheep or anything like that? I'm not so familiar with the auroch, which is this cow, but there was an effort in Poland, which I think is the same, which is with the tarpon horse. So I talked about the Przewalski, so that's the eastern wild horse that was never domesticated. But on the west of the Ural Mountains, there was a tarpon horse. And the tarpon horse, the last individual, the last that died was in the 1700s. So that's a truly wild horse from the west in Europe. And so that became extinct. And there was an effort by a geneticist in Poland to bring it back. But the way they did it is basically go to the forests and try to identify individuals that look like the tarpon horse, and then breed them back. Now, was that the actual tarpon horse? Probably not. Probably there was a mixture of all the breeds. It's an effort. It's called a new tarpon horse. What we're trying to say is, if we actually had the samples, in the year 2000, leading to 2000, it took us 10 years to sequence the human genome, right? Now we can sequence the genome in 1 day, right? You can literally have a baby, and the next day you know their genome to save them. Right? So imagine what we can do 50 years from now. But what we really can't do 50 years from now is have the information to make what really existed from before. So that's where we are. But yeah, those are efforts that. There's. At some point, those work. For example, the Florida panther, you put a Texas puma, and that saved the population. Was that fair to do? They're actually two different species. Well, actually, probably was fair to do, because the range of the Florida panther and the Texas puma was actually overlapping at some point. It's only fragmented because people invaded it, right? So in a sense, that rescue was very, very, very good thought. And maybe they could have done the same with the northern rhino, right? Maybe, because they were close enough, and there probably was a level of intermixing at some point. So those are the kinds of calls that conservation genetics have to do. But having the samples can only help. Yeah.
Thank you very much. Really appreciate it.
Thank you. Thanks a lot. Yeah.